Thank you so much, Rhona. And I'd like to just thank the Raine Study team for inviting me to talk this afternoon. It's a great pleasure to be here, despite being on maternity leave. So if I'm a little sleep deprived, please be kind. But I'm going to talk today about some of the work that we've been doing in phenomics, and in particular in the context of population studies and what that means in the quest towards precision medicine. So what is phenomics? Essentially, phenomics is the approach to studying the interaction between our environment and our genetics. It's a way of looking at biospecimens and trying to tease out all of the genetic components that lead to our disease risk alongside our lifestyle contributions. So we can do this by studying biospecimens that are readily available, such as blood and urine, and ideally in large-scale population cohorts where this allows us to look at trends in assessing early biomarkers of disease, biomarkers and the ability to detect diseases much earlier, and the ability then to try and develop stratified ways to treat people towards a more precise way of looking at individuals rather than treating everyone the same. So essentially what we're looking at here is all of the contributions that we're exposed to throughout our life course. Those are environmental factors that include things like our diet and nutrients, medications, any pollutants that we're exposed to, environmental chemicals, which is a particular interest of mine, as well as our microbiota. So all of the microbes that live within our systems as well have a huge influence on our metabolism and the metabolites that are produced in our biospecimens, and how that interacts with our genetic profile as well. So all of the genetic contributors to our metabolism, metabolic pathways, and this complex interaction is really what we're studying by looking holistically using the phenomics approach. So within the multi-omics workflow, metabolomics or metabolic phenotyping, lipidomics, these names are used interchangeably, sit at the bottom of that cascade, very close to the biological phenotype, which gives us great potential to study these metabolites and how they are reflected and how they're reflecting the biological phenotype. It really represents all of the small molecules and metabolic pathways that are driven right from the top of that workflow with our genomics. And actually combining all of these omics together is a truly powerful way of looking at a systems biology approach to health and disease. At Murdoch University, we have a world-class facility in actually undertaking these phenomic studies. This is the Australian National Phenome Centre and Centre for Computational and Systems Medicine, which houses a huge array of analytical technology designed to study biospecimens, to deeply phenotype samples, and get very specific, accurate and precise measures of all of the metabolites that we're trying to capture. These analytical equipment are very sophisticated, and there's very few laboratories that are set up as we are to do this on such a large scale. And that's one of the true values of the operation that we're running, is that we're really now able to tap into population cohorts and longitudinal studies, which give us this magnificent window into early life effects on our lifelong health. What we're trying to do, though, is very complex. We're trying to measure all of the small molecules that are in our biological samples. That's a huge, diverse array of biochemical compounds across a huge spectrum of concentrations. So being able to do that in any one single assay is impossible. And what we do is we devise these methods to look at particular windows of metabolic pathways. So we have lots of different methods and assays to try and holistically capture the whole metabolome. And of course, then there's a whole dark metabolome in the background, which has not even yet been characterized. So there's many, many chemicals and metabolites in our samples, which we don't even know what they are, how we can measure them. We use an untargeted approach to do that to complement our targeted quantitative assays. In terms of our targeted quantitative methods, I'm going to focus mostly on two today and give some examples of how we've applied them in different studies. And that's our lipoprotein profile and our lipid profile. And these chemicals are known and are measured by standard clinical methods to give us very detailed insight into diseases, inflammatory conditions, and disease risk. But the panels that we have are able to offer a much more detailed and intricate picture of the links between these molecules and diseases. So the lipoprotein profile will quantify over 100 different lipoprotein subfractions, and our lipid assay over 1,000 lipid species. So very, very large sets of data which give very detailed picture of the metabolic pathways involved. And commonly these are applied to standard control and case studies. So where we've actually got predefined recruitment cohorts of a healthy control group and a particular disease group. And that enables us to look for associations between metabolites and disease outcomes and the potential to develop biomarkers. But much more powerful is applying these tools to population studies. In population cohorts, then we're looking at real-world people going about their daily lives with no intervention, and then we're actually retrospectively mapping the biochemical fingerprints that we're able to generate with the onset of diseases and symptoms before we've actually collected those samples. So this retrospective picture is really, really powerful now in looking and helping us to determine early biomarkers of disease and the onset of disease before symptoms occur, as well as starting to look at the causal pathways, which is obviously very powerful in understanding more about the mechanisms of disease, as well as trying to develop new therapeutics and precision treatments to different diseases. And we've been doing this already with some of the WA population cohorts. So the Busselton Healthy Aging Study is one where we've applied our lipoprotein and lipid profiles to a subset of the population cohort, so about 2,000 participants. This is also a long-term and ongoing population cohort within the WA community. Angled towards a slightly older population group than the Raine study, so very complementary. And what we've done here is we've applied our methods. This is some of the work that one of my PhD students, Dr. Alana Grant St. James, has been working on. And she's been looking to kind of disentangle this lipid, the plasma lipidome alongside body composition and what that can tell us about our disease risk, particularly in the context of cardiometabolic disease. And what she was able to determine from performing this lipid and lipoprotein analysis. So what Alana was able to show by doing this detailed lipoprotein and lipid analysis is that there are these very unique characteristic lipid profiles which reflect our body composition. Of course, we all have different body compositions, and there are certain body compositions and the way that we store our fat and excess fat that leads or render us at a higher risk of developing diseases. And what Alana has shown that those people from DEXA scan imaging that held their fat around their visceral and trunk areas were at a much higher risk of cardiometabolic diseases, which was reflected in these lipid profiles that were demonstrating those high risk pro-inflammatory markers. And what we were also able to determine was the sex differences that were very distinguishable between these profiles as well. And in particular for the females who held fat around their lower limbs and their legs actually suggested a more protective mechanism against cardiometabolic diseases. So good news for all of the ladies out there who carry weight in those areas. Another area we've been working on is in endometriosis. So a condition which is very difficult to diagnose, takes a long time for women to get diagnosed with endometriosis and does require quite an invasive laparoscopic surgery to do so. So this is work that we've been doing in collaboration with the End Origins Biobank, driven by King Edward's Memorial Hospital. And what we've done here is actually applied the same methods, those lipid methods, to plasma samples from individuals with endometriosis compared with those that have got no endometriosis diagnosis. And this plot here shows very clearly the separation between the controls, the blue dots, and the pink dots, which are those individuals with endometriosis. There's definitely some overlap in there, which goes to show that perhaps it's not quite as easy to characterize the disease. It's a big spectrum of disease, it's very heterogeneous. And those presenting with symptoms perhaps don't have endometriosis and vice versa. There may well be people out there who aren't diagnosed but do show signs of endometriosis. This eruption plot here shows us the actual lipid species that are responsible for creating that separation. So the features in here, which are very highly significantly associated with endometriosis, those over on the far left-hand side, a lot of triacylglycerides. So this is now showing us a little bit more about the mechanisms and the pathways involved in that disease. Again, helping us to learn more about the mechanisms behind the disease will be really informative in developing new treatments as well as diagnostic tools. And one of the really interesting things we're able to do with this cohort is actually project onto that plot individuals that didn't they weren't able to receive a diagnosis because during that clinical surgery, there was no sign of endometriosis found. They presented with symptoms, but they didn't get a diagnosis. We presented their profiles onto this plot, and you can see that those open circles sit either within the endometriosis cohort for 2 of the examples, and one is sort of in the middle of that boundary. This shows that certainly for those 2 that sit with the pink dots, they have biochemically the same fingerprint as someone with endometriosis. So certainly there's evidence there to suggest that they have the same profiles of endometriosis, despite that not being found during the surgery. And that's very plausible because endometriosis can be found anywhere within the body. During that diagnostic surgery, it's only really looked for in the pelvic region. So it does go towards showing that these systemic tools have a much better ability to maybe predict and help diagnose complex diseases such as endometriosis. Taking that a little bit further, we were actually also able to distinguish between mild and extensive types of the disease. The disease has many different phenotypes. As I mentioned, it's very heterogeneous and it can be broadly categorized into minimal and extensive. The lipid profiles reflected that degree of severity as well. And we were also able to see that that was largely driven by these triacylglyceride species. So the same species that were very much linked to endometriosis compared with controls also start to show that they're very closely linked to the severity of the disease as well. And again, we're able to project onto that plot the ones which didn't receive a diagnosis, and you can see that, a number of those do sit within the endometriosis phenotypes as well. So not only are we able to determine whether they have endometriosis or not, but actually perhaps how severe it is. Now, in terms of longitudinal sampling, this gives us a real valuable approach to actually looking at the trajectories and recovery trajectories in many cases of different insults and injuries. In this case, work that's been done by one of our postdocs, Dr. Monique Ryan, and she's been working very closely with Fiona Wood and the Fiona Wood Foundation with the Pediatric Burns Biobank, and actually also using the Origins Biobank as a control cohort for these samples. So what Monique's been able to do here is do lipid profiling on children who have had a burn compared with controls who haven't received any burn. And you can see here that for both males and females, there's a very clear distinction between the 2 groups, healthy and control, which don't have a burn. Again, you can see the lipid species, or perhaps you can't see that terribly well. But during that acute inflammatory phase, right after the burn, it's very clear that there's a huge systemic effect on the lipid profiles, which we can pick up very easily. Interestingly, Monique's done some work on some follow-up patients, and this is over a year after injury, where the burn's largely healed and resolved and there is no physical sign of that burn any longer. But actually, systemically in the plasma samples, we're still seeing effects that have been attributed to the burn injury. And it is known that participants who have had a burn injury in their early years are a much more higher risk of developing certain diseases later in life, cardiometabolic diseases in particular. So this now starts to give us a bit more information as to perhaps why that is. Why is it that these individuals don't return to their kind of lipid profile and what that looked like before their burn, but it's actually been in some way extensively and irreversibly changed. And also, again, in this case, we see differences between the males and the females as well. So in these children, the female participants in particular seem to have a much more sustained difference in their follow-up time point compared with the males, suggesting that perhaps the females don't recover quite as quickly as the males do. Now, a lot of our work over the past few years has largely focused on COVID-19. At the time, that was obviously one of the biggest health risks of our time, and we pivoted a lot of our focus to looking at COVID-19. And since then, the long effects of COVID-19 and long COVID, where we see that participants are still reporting symptoms long after the acute infection has been resolved. So we've done a lot of work in this space as well to not only look at the acute infection, but also what are the ramifications and the long-term health impacts. And we've been able to do this through great collaborations around the world with researchers and other phenome centres who are running harmonised assays. I'll just show a couple of examples here. So again, this is all the lipids that we've measured and showing the diagnostic potential of these lipids in terms of acute infection of COVID-19. This tool was very easily able to discriminate just based on 6 lipids that we whittled the huge panel down to, very highly discriminatory between the healthy non-infected participants and our COVID-19 infected participants. very strong ability to predict those. So in terms of diagnostic context, it really shows the ability to translate some of these tools and discovery research that we're doing into clinical diagnostic tools. And again, not only were we able just to distinguish between control and infected individuals, but also, again, looking at the degree of severity of symptoms which can present. So just looking at the initial time point when individuals presented to a hospital to get sampled, we were able to put them into risk categories to demonstrate, just based on that one single time point and one sample, actually the difference in these lipids across the different degrees of severity that those participants resulted in. So just from that very early onset of symptoms, we could tell which people were going to become more severe than others. And again, that shows the great potential of being able to use this type of technology to stratify patient care and treatment. And what we're working on now with our collaborators is looking at the effects of long COVID-19. So again, after the resolution of the acute infection, a lot of people are still rendered with these symptoms, and perhaps not only if they're not presenting with symptoms, certainly their biochemical profiles have been changed and have this persistent profile which is consistent with higher risk of diseases such as cardiovascular disease, diabetes, obesity. So mapping these metabolic risk spaces is hugely valuable. And first of all, we need that foundation of the healthy population to work with. And that's where studies like the Raine study can really be such a valuable resource in determining where we are to start with before we have any insults or any of these effects in our lives. We occupy this metabolic space, and then whatever disease or insult might occur, whether it's an infection or a burn trauma, might render us in a more inflamed state. And then a lot of people will return to a healthy baseline, but a lot of people might not, and they might end up now in a higher risk area for diseases such as cardiovascular disease, diabetes, obesity. And really understanding the population health and population phenotypes and profiles gives us the ability then to study how these different insults affect different individuals. And I know I don't need to tell this audience the value of the Raine study, but in terms of a longitudinal population cohort, which is multi-generational, has got such a wealth of data that goes along with those biospecimens, it's such a valuable resource to be able to apply this type of technology to really start to unravel the origins of disease, and looking at, across the life course, all of the things that can happen and how they will influence our health and disease state going forward. So to summarize really the whole purpose of what we're doing and why we're trying to do it with large population cohorts like this is to determine the origins of disease. In doing so, we're able to really understand and get a very detailed picture of the influence of genetics, environment, and lifestyle to be able to predict future health outcomes. In doing so, we are also developing translational tools for clinical impact. So whilst this is very much a discovery-based science, we do want to be able to translate this to real-world impact and be able to develop tools which are able to be deployed in a real-world setting. So actually scaling down some of the sophisticated tools that we have available and working with industry to do that, making sampling much more accessible, and remote sampling in particular is a very attractive tool for doing that, particularly in Australia. And overall, the ultimate outcome, obviously, is to hopefully prevent and be able to predict these diseases so that we can prevent them much better towards precision treatment. And of course, that's got many, many benefits, but not only in reducing the incidence and prevalence of diseases, but also in improving the health outcomes for individuals and making sure that we're actually able to provide inequitable healthcare for everyone. This has been a truly collaborative presentation with lots of input from my colleagues, both at the Australian National Phenome Centre and Centre for Computational and Systems Medicine, also for our collaborators at the biobanks and funding sources as well. So thank you very much to you all. Thank you.